हेलो। एक दे।
So it will now you can if you want you can speak.
Can you hear me? Sunita, can you hear me?
Hello. Hello, ICGB, can you hear us? Ma'am, your voice is not audible. I think your mic is muted.
Mic is muted. Can you hear us now? Unmuted.
हेलो। किनका उनका? हेलो।
We can hear you very clearly.
Good
I'm
afternoon.
Sharma. Good afternoon. So, I think there may be some issue with the room. संजय, आवाज तो आ रही है वहां से आईसीजीबी से? I think some. I can hear you very clearly.
Thank you, ma'am. That helps.
Just send a message. Send a message to Sanjay or Suraksha.
Yes, ma'am.
Now speak now.
Sunita?
Yes, ma'am. I can hear you now.
Okay.
And all other participants are online. I mean,
So we have some who have joined on Zoom, and we are also broadcasting it on our YouTube channel and also on DBT channel. So I guess we will have a audience.
the four speakers.
So we have Dr. Aishwarya already online.
We
And
can't
Dr.
see.
Lakshmi.
Okay.
Two of them.
Okay. But
I'll call them again, sir.
because they should join, no? Otherwise,
Yes, sir. I'll do that, sir.
let all four join.
If you want to speak something. Acha, can you come here?
A very good afternoon to everyone who have joined us today for this event. On behalf of Department of Biotechnology and International Center for Genetic Engineering and Biotechnology, ICGP New Delhi, I, Rita, welcome you all to today's program. Globally, we celebrate International Women's Day on 8th of March to honor the achievements of women in various fields. The theme of today's program is Women Excelling in Science. The celebration acknowledges the women personalities who have excelled in their area of expertise. Now, I request Dr. Rita Maheker, Director, ICGP New Delhi, to give the inaugural address.
Good afternoon to you all and welcome to the joint program by ICGB along with DBT to celebrate the International Women's Day. It is a global day to celebrate achievements, as Subira said, and also to remind us to achieve the gender balancing and accelerating women empowerment. According to the known data, 50% of the undergraduates in life sciences are women, which is perfectly alright, but subsequently that number keeps reducing, and it is about 30% of scientific researchers are women, and further if you go and look at the faculty positions at professor level, it is less than 25%. So this trend seems to be actually alarming in the sense that if you want to have equality among men and women, it should actually be achieved at all levels, and that should be our goal. So it is quite evident that we do have increasing number of women as a workforce in many spheres, but it still is not adequate in terms of scientific areas, and that is something that for us who are involved in scientific research should be very conscious. We do have a large number of students coming for PhD, but eventually in our institutions, number of faculty who are working at senior level is significantly less. So we should, for one more year, we should actually resolve ourselves that we should actually increase that number, and to increase that number, it is not sufficient that management only is putting efforts, but also women scientists across the fields also should put efforts. ICGB has actually done tremendous work in this context. Particularly, we have been having a large number of programs where we focus on women participation. Very recently, ICGB has started a program called V-Star, and we have been promoting women scientists from African countries to come and work in ICGB Cape Town, ICGB Trieste, and ICGB New Delhi. And that selection process has just been begun. We have interesting proposals between ICGB and DBT to actually enrich women participation, particularly from the poor countries in the neighborhood, to come and work in New Delhi component of ICGB. So I'm sure slowly all these things will materialize. We will international our women scientist participation in our activities, as well as we actually enriches. So today I think we have galaxy of star women performers going to give presentations to us. So let me welcome you all again for this celebration, which is brief, but I think everybody is actually going to give us a pointed suggestions as to how to proceed further. And I'm also happy to see Dr. Rajesh Gokhale and other colleagues in DBT to be participating in this program. So let me welcome you all again and thank you for for giving me opportunity to speak to this audience.
Thank you, sir, for your kind words. I now invite Dr. Suraksha Devan, Scientist E, Department of Biotechnology, Ministry of Science and Technology, Government of India, to brief about the two women-centric schemes, Biotechnology Career Advancement and Reorientation Biocare Program, and Janki Amal Bioscientist Awards. Dr. Devan.
A very good afternoon to one and all, and welcome to this occasion of International Women's Day. Thank you, Secretary Deputy, for gracing this occasion of celebration of Women's Day with your kind presence. Women scientists have been playing an important role in scientific growth of India. Their outstanding contribution to science and societal growth has been remarkable. The department has the mandate of promoting women in science and science for women, and it is also very important that we acknowledge contribution of women scientists who have done excellent work. In order to recognize the outstanding work done by the women scientists who are in permanent positions, outstanding contribution in the areas of biosciences and biotechnology, the department had initiated National Bioscience Awards for women scientists in the year 1999. These were renamed as Janaki Amal Awards in the year 2019. Padma Shri Dr. Janaki Amal was a pioneering biobotanist, Director General of Botanic Survey of India. There is a small flowered variety of Magnolia plant named after her, called the Magnolia Cobalt Janaki Amal. The awards are given in two categories, senior category and young category. In the senior category, senior women scientists are awarded for lifetime contributions and excellent research in the country. In young category, award is given to women scientists below the age of 45 years for outstanding contribution in research with potential for application, product and technology development. This carries cash prize, citation, gold medal, as well as research grant also in the young category. So far, 72 women scientists have been awarded since inception. Today we have with us Dr. Aishwarya Lakshmi and Professor Shamishta Banerjee, the awardees, who will talk about how this award brought a change in their careers. With the aim to promote women in science and in attempt to enhance the participation of women scientists in biotechnology research, and had launched BioCare program for women scientists in the year 2011. The purpose is to give an opportunity also to women scientists who are not in regular positions or had a break in their careers to get their first independent research grant, including fellowships, and thus the chance to get back to the mainstream of science. Through this scheme, we have been able to support around 360 women scientists with their first research grants. Many who were unemployed at the time of getting their respective biocare projects have now got permanent employment in various institutes, universities, and industries. More than 400 papers have been published, 11 patents have been filed, and one international patent has been granted recently, which is remarkable as these are from their own companies. Last year on this occasion, one biocare awardee who got permanent employment in an Indian Central University had spoken, and one who became co-founder and CEO of the company had shared her journey. Today we have Dr. Anandi, who will talk about role of biocare in her journey, right from getting her first independent project to foundation of her own company. Through this, she has created employment not only for her but for many others. We also have Dr. Lakshmi Prasanna, who has recently been granted a US patent from her biocare project. They will share their success stories before and after getting their first independent biocare R&D project and how it made impact on their careers. They will also talk about how their novel ideas saw light of the day through this scheme. Besides these, it is also important to acknowledge work of great women scientists of India who started their scientific journeys in 1900th century and made a mark in STEM area. And what is better than to showcase their stories, journeys, and thus inspire the young minds of India. In order to do so, an exhibition through posters will be put up in colleges from next month with aim and hope that this exhibition will motivate young students, that is the building blocks of India, to take or continue science as a career and contribute to socio-economic growth of India. This was planned 2 years ago, but could not be executed due to COVID-19 pandemic, but colleges are not open now. Thank speakers, scientists, principals, lecturers, teachers and students from various institutes, universities and colleges who have joined this event. Hope all of our efforts, today's event and talks will motivate youngsters to take science as a career, help women researchers and scientists who have just started their careers or those who wish to revive their careers, and thus help in creation of a strong generation of women leadership in science and technology in future. Thank you.
Thank you, ma'am, for the wonderful summary of the journey. Now I request Dr. Sanjay Kumar Mishra, Senior Advisor, Department of Biotechnology, Ministry of Science and Technology, Government of India, to give his address. Dr. Mishra, sir.
Good afternoon, everyone. Once again, on behalf of the Department of Biotechnology, I would like to extend my warm greetings on this International Women's Day, which is celebrated on 8th of March every year. Now, on this day, I think this is the time for all of us to reiterate how important role the women have been playing and are playing into all walks of life and all spheres of life, including science and technology. I would like to just give you, just brief points that women in general are so important into the education sector and into the science and technology sector. First, if you take the Sustainable Development Goals, there are a number of goals, but goal number 5, which is specifically talking about the promotion and support of the women into the education and providing them all support. Now, if you do, just a brief analysis, you will find that if we support number 5, if women are being empowered, all other sustainable goals will be much more easier to achieve because this is a horizon presence into achieving all other goals. So this is not only important for the benefit of women, but in achievement of overall sustainable development. Now, second point is sometimes we think that by promoting women scientists, we are helping them or we are helping individual women scientists, whether they are into the education sector or into workforce. Now, if you read the several research papers and analysis, which has been done by the researchers and also confirmed by the recent studies of the World Bank and IMF, they have shown that whenever the participation increases into workforce, I'm not talking about science and technology, but in general, then that workforce is the increased workforce having a diversity, inclusion of women, that increases the productivity, which increases, if it's a private company, which increases the profit, and as a result, more money is available, and that increases the wages of all the employees of the organization. So in other words, there is a case where the empowerment and participation of women at all the level is not only helpful to the women, but to the overall organization, overall nation building. This is the second point. The third point which I would like to again to tell is that if you look at the data of last 15 years, especially I look into two boxes as a Department of Biotechnology person. One is the education sector, right from primary school to middle school to college to PhD level, and second is the workforce. So you will find that considerable increase and improvement has been made into the education sector. Like if you look back about 20 years back, the gender parity index or the participation of the girls into the school level was I think 0.9 or 0.8. That means out of 100, we have huge deficit. Over a period of time, that I'm happy to state here that the last survey, the survey which is done by the Ministry of Education, that is showing that a very healthy participation student at school, college and university level, and that is close to 1. And especially if you look at the higher education sector, again you will find that the gross enrollment ratio of the girls and women into the education is again very healthy. Of course, there are certain deficits, discipline-wise, theme-wise, still there are gaps which need to be fulfilled. However, in last 15 years, I can say the trend has been very positive. Now, the second point in the same theme, which I wish to sensitize, and not only with the government agencies, but to the industries, but that the concomitant growth or improvement has not been seen into the workforce. Because if you look at the participation of the women, if I look at the data published by the Department of Science and Technology, the last one, the latest, the last one, I can't remember the years, but it was 14.7% of women into SNT workforce, and the latest one is saying 16.5. So that has been a very, very, very minimal, improvement. I would be happy if this goes to the 20%, which is the Asian average, or it goes to the 28%, close to 30%, which is the global average. So here is the gap. So the message which I am taking myself as an officer of the DBT, I also wish to remind that we will work hard to promote and to enable, to create the environment so that not only the education in terms of the various fellowships, scholarships, and postdoctoral fellowships, but also create the avenues for women scientists to be absorbed into the workforce system. Once again, I won't take much time because I'm also waiting to listen the women scientists and women achievers from the DBT and from the various awards and fellowship who will tell about their own journey, and I would like to take lessons to be learned from them and recommendations suggested by them. So finally, on behalf of the Department of Biotechnology, I wish to reiterate and confirm we will do our best possible to support all the girls and women at student level, at HRD level, on all the schemes of the DBT, wherever possible, to provide them support, which we have been doing in the past. But nevertheless, if there are scopes which can be improved, if there are certain areas, we will be more than happy to further reinforce and take the journey to the further, so that both the whether the SDG goals are achieved and women participation in education as well as into the workforce sector is achieved. Thank you very much.
Thank you, sir, for your kind comments. I now request Dr. Rajesh Gokhale, Secretary DBT, Ministry of Science and Technology, Government of India, to address the item.
Thank you, Subira. Thank you all of you for coming here today on this wonderful occasion. I first of all extend my warm greetings to all of you on the International Women's Day. I think it's one of those days that we all love to celebrate, primarily because the way I look at it is because all our life we are surrounded by women in different capacities, and all these incredible women actually do such an important role in our life. There is no reason why you can't thank every day, but on a special occasion it's even bigger and higher. So thank you again, all of you, for making this life such wonderful and gregarious. I'm indeed honored and elated to grow with all of you actually in my life, and this has been an incredible journey, and I must say it's a wonderful occasion to say that actually. I think the way I look at it is gender is part of one's identity actually, right? I mean, I think this is nothing that you can change or something like that. I think what we can change is these gender specific changes should not demarcate boundaries both in societal and cultural or professional ways actually. There is no reason why we should demarcate those activities, and that's what my real motivation in all these is. We should have freedom to the way everybody wants to work and the way everybody wants to express themselves and carry forward themselves actually. I think that's been the most wonderful part of what Department of Biotechnology has been doing. In fact, if you look at Department of Biotechnology's history, it's one of the unique departments where even 2 secretaries were women secretaries of the 5 secretaries, in fact, have led Department of Biotechnology from a point of, in the past several years, and they've done it with great finesse, and all of us, would acknowledge that their contributions have been enormous and immense to the growth of. So I think it's wonderful to know that we work in an environment where the women are already given lots of, they are the one who are giving us directions and giving powers and giving activities to be able to look at. However, some of the data, as several of you collected, I also looked at Google and other places to try and find some interesting data to look at. And one of the interesting data that I found on the World Bank site was rather interesting actually. And this said that about 43% of the women in India, in fact, complete their graduation in STEM areas. And I thought this was rather incredible actually, if that's a number, because that means that there are more and more people who take up to the STEM areas of research, STEM areas for their graduation. And this is even higher than almost all developed countries. So I got this number saying US is about 34%, UK is about 38%, Germany is 27%, and France 32%. And I think in India it is about somewhere around 43%. So it's incredible that, people take up to the STEM area of science, technology, engineering, and math, in these areas of research for their activities of carrying out future. Unfortunately, even though despite this, the number of R&D personnel that works in various posts, I think broadly it's about 3 lakhs or people, if you look at from what you look at from an R&D setup in Indian perspective actually that works. Unfortunately, the number of people who survive as a long-term in the STEM area is still about 15% of them, I think. And I think this is something that is a very worrisome factor from an administrative point of view or from a point of view of the way we look at gender balance in our society and system, I think. And I think that's clearly need to be corrected, and that clearly need to re-looked at why is it that scenario happens actually, and is it there, therefore how do we correct it? And I think that's what Department of Biotechnology, as Dr. Sanjay Misra and Dr. Suraksha Diwan both mentioned, is that in fact it has several programs that are associated specifically to bring people to be able to get back if they have breaks in their careers, how to bring them back again and re-energize and refocus and get back into their own values of what one can look at actually. And obviously there are other programs that one has to do this at the school level, at the college level, at the entry level. And clearly we need to bring out more and more motivational aspects of lives that actually get some of these people to do well together. In fact, if you look at sports is one of the areas where women's sports personals have been actually doing better than men counterpart in almost all disciplines of sports. If you look at Liverpool cricket, which has a long history, and that cricket history is still very small, but nonetheless they were also semi-final. They were in the final last year of the World Cup 2020 as well. So clearly, there's a tremendous amount of environment that one should actually feel excited about being part of this ecosystem and be able to carry out actually. So I think, let me just say that, I think what department has decided to do, in fact, to be able to, carry forward some of these fellowships and awards that are being granted, is to double their numbers from this year onwards. And I'm really thankful for all my colleagues to be able to do this. I think this is both for Janki Amal fellowships for young women as well as for the senior women actually. And we would also enhance their fellowship amounts that we will provide them to carry forward their work. So hopefully this is a small way, but I think it will keep changing, and if we keep moving ahead as more and more money flows into the department, actually we will continue to empower and carry forward these activities. So this is a small, I understand this is small for the total size of the country, but there's always a beginning to be able to make and be able to carry forward actually. Finally, I really want to again acknowledge all the past and present women people in Department of Biotechnology, because that's where I am present here for, and I think there are lots of wonderful women and I think I really admire them for their dedication, for their commitment, and for all the complete professional ethics that they carry forward to. And I think they have tremendously contributed to this department's success. As I have said again, reiterate the two secretaries by themselves have been women in this part of things, in this activity. I think with this I will just stop, but I'm very looking forward to listening to some wonderful, a small talk of about themselves and about it, and where are the other possibilities that we all can inculcate some good ideas and practices. And I really look forward to the six of you whom you're going to speak after me. And I think I'll just stop here, but say thank you very much again. And this is very, very important for us that we actually celebrate successes of people actually in variety of forms. And I think thank you very much again for coming. And thank you, Dr. Salunke, for hosting this today's meeting. It's a very, very important activity that you are doing over there. And I'm really grateful from Department of Biotechnology is grateful for this activity. Thank you.
Thank you, sir, for your kind words and guidance. We now move to the talks from women researchers. Our first speaker is Professor Rita Maheker. Professor Rita's area of specialization is in oncology. She is Professor and Scientific Officer, retired Homi Bhabha National Institute and ACTRAC, TMC, Mumbai. Presently Director of Samartha Krupa Life Sciences Private Limited, Pune. She is Fellow of National Academy of Science, India. She is a senior scientist and has been a mentor to many students. She serves as an expert on many committees and panels associated with the department, including that of DBT Biocare, committees at BIAC, at Ministry of Health and Family Welfare, ICMR, and DST's WAS in Life Sciences Committee. Professor Rita.
Yeah, thank you very much for the kind introduction. And I would like to first of all wish all of you a very happy International Women's Day. Can I have my slides? I think I've sent my slides to you. Can you project them, please?
Yes, ma'am.
Yeah, thank you. So first of all, I would like to thank Dr. Suraksha Diwan for inviting me, and I'd like to congratulate her for holding this half-day event on International Women's Day. Next slide, please. So very often I'm asked, how was I motivated to be a scientist? And when I think back, I think I was just fortunate to be born in a family where women were encouraged to take up challenging activities and to pursue a career in science, a career. And I was really very fortunate to have two of my grandfather's sisters who were highly qualified in those days. Both of them had traveled abroad. One of them had gone to UK to get a MRCAG degree, and the other one was in Edinburgh doing a PhD under a very famous embryologist, Dr. W.H. Waddington. And so they were like role models for me. Also, my mother was one of the first lady pilots to get a private pilot's license, and all this was in the late 40s, early 50s, remember, when women were not even allowed to get out of their house. So this was something that probably led me to pursue a career in science. Next slide, please. Can I have the next slide, please? Yeah, thank you. So after doing my PhD, I was selected in a UNDP UNESCO course in modern biology at Biological Research Center in Szeged, Hungary. And there I was fortunate to learn the then latest technology of hybridoma, making monoclonal antibodies. And that was the time when Kohler and Milstein had just got the Nobel Prize for this hybridoma technology. Later, when I returned to India after those 2 years in Hungary, I joined Cancer Research Institute, and I helped Dr. Damayanti Shah at the Radiation Medicine Center to make monoclonal antibodies against thyroglobulin. And this was again in the early 80s. So, you know, things were very different at that time. We didn't have any, we had to do everything manually and handle so many 96 well plates, nurture the cells, screen the supernatant for antibodies. And so for all this, you know, it took a lot of time. I don't think I spent a single weekend at home. I was always in the lab. Then I worked at ACTREC, which is what was earlier called as Cancer Research Institute, for more than 35 years. I was again fortunate to work with Dr. M.G. Deo, who encouraged me to initiate projects in futuristic medicine such as gene therapy. And you know, this was 30 years ago when people in India hardly worked on gene therapy. And in fact, some of my senior colleagues tried to dissuade me. They said, why do you want to do gene therapy? This is something that's going to be so expensive that no one in India will be able to afford it. But you know, the picture has changed completely now. I also made a transgenic mouse expressing a gene ectopically in the skin. Again, I was sent for training to US by Dr. Dev, and when I came back, this was again in the late 90s that he made the first transgenic mouse here. After superannuation, I joined a startup company, and I'm now working on cell therapy for patients with back pain due to herniated intervertebral disc, and we hope to do clinical trials soon. Next slide, please. Okay, so we know that women face several challenges in professional development. Women are constantly striving to come out into their traditional homemaker and mother roles to professional career woman roles. And some of my friends and colleagues have really managed to do this very successfully, and they manage both fronts very well. So, you know, this should not deter any of the young upcoming women scientists. However, we know that pregnancy and related ramifications are still primarily women's concerns in some cultures, and continue to slow down the growth of many women professionals, in addition to other societal perceptions in India. In fact, I remember I had a very bright young girl who joined my group for a PhD program, and I was really happy that she had joined me. She was a really intelligent girl. But soon after she joined, she got married, and I had no problems with that. But she had a tough time handling both situations. Her mother-in-law would want her to go for all the religious ceremonies everywhere, every day, and she hardly gave any importance to the research that this young girl was doing. So unfortunately, the girl had to quit her PhD program. So constant attempts are going to steadily improve the gender climate in several aspects. And there is an increasing number of medical graduates and scientists who've done really well. And we know there are two examples from DBT. We know Dr. Manju Sharma and Dr. Renu Swarup, who reached the top position of power at the Department of Biotechnology. So even then, I think women have to be at least 10 times better than the men to reach such positions of leadership. Next slide, please. Single-mindedly pursuing a career is easier for men, definitely, than for women, as evidenced by the pattern of work and working hours, time spent on domestic family chores, and conference attendance rates of male versus women. Pregnancy and maternity leave are still viewed with much bias by most employers. Women have to walk a tightrope to balance family, clinical care, and research pursuits, in addition to maintaining and advancing professional skills. So efforts like fellowship and mentorship options, support for women after maternity leave, both flexibility in education programs and accessible care development, skill development programs, and leadership workshops is the need of the hour. Better childcare facilities at workplace and conference venues are considered helpful for advancing women's careers. So next slide, please. We know, we've just heard about some of the initiatives that DBT has taken for to encourage women to return to their career after a break. In addition, there is Women Scientists Scheme WSA, which is run by Department of Science and Technology, and the Kiran Scheme from the Department of Science and Technology, and of course BioCare, which is what is holding this meeting. Next slide, please. So I would like to end with these words from one of the most admired women scientists who has got 2 Nobel Prizes, Madame Marie Curie, who said that life is not easy for any of us. But what of that? We must have perseverance and above all confidence in ourselves. We must believe that we are gifted for something and that this thing, at whatever cost, must be attained. So all the young women scientists in the audience, I must tell you that you must persevere and have confidence in yourself, and you will be successful. Thank you. I'm done.
Thank you, Professor Rita. We now move to our next speaker, who is Dr. Aishwarya of National Institute of Plant Genome Research, New Delhi. Her research interests include understanding the interaction between sugar and plant hormones and its effect on plant growth, development, and stress responses. She is recipient of Janki Amal National Women's Bioscientist Award from DBT 2015, elected Fellow of National Academy of Sciences APR 2016, elected Fellow of Indian National Science Academy 2021, elected Fellow of Indian Academy of Sciences 2021, along with many other international travel grants. Dr. Aishwarya, please.
Very good afternoon, everyone. So thanks a lot to DBT as well as ICGP for giving me opportunity to talk on this wonderful occasion that is International Women's Day. So I congratulate you all for organizing such a beautiful function. And I would like to share a few of my slides. I'll show you the work I have been doing at NIPGR for the past 16 years and what brings me here on this platform. So I'll just share my screen and show the work I have been pursuing. So can you see my slide?
Dr. Aishwarya, we can see your slide. You need to put it in projection mode, please.
Yeah. I have some problem projecting it in the projection mode. Yeah, can you see it?
Yes, please.
So basically what I do at NIPGR is to study how does sugar signaling, it controls plant growth, development and stress responses. So sugars, as we all know, it is produced by photosynthesis, and once it is produced, it not only acts as a source of energy, but it can also act as a signaling molecule. So in my lab, I have been trying to decipher how does sugar acting as energy or a signaling molecule control plant growth development as well as the stress responses. So sugars once produced, they can either be perceived by hexokinase dependent signaling, independent signaling, or they can be perceived by two kinases, which are two important kinases. One of them is TOR kinase, which promotes growth and development when there is ample energy in the system. But when the energy is less and the plant is stressed, then SNRP kinases, they get activated. And what they do is to block TOR kinases, and they block growth and development, and they induce stress responses. So when I joined NIPGR, there was hardly anything known about how does sugar regulate plant growth and development. So I did very basic fundamental work, and I tried to understand how does presence of glucose affects various growth parameters in the plant. So I put the plants in various growth conditions, and I could actually see that glucose profoundly affects plant growth and development, not only in terms of root length, lateral root formation, root hair formation, but surprisingly also the gravitropic responses. So glucose could alter all the responses. So then I thought maybe glucose does it via altering some of the hormonal responses, because all these responses, all these features are also governed by several hormones. For that, I conducted several experiments in terms of like physiology, molecular biology, cell biology, and I could figure out that glucose interacts with various hormones such as resveratrol for controlling the directional growth, or with cytokinin to control the root hopping response. So they were very beautiful responses in which glucose and hormones, they could interact with each other. So later on, we also figured out that glucose also controls the number of lateral roots as well as their angles, angle of emergence. So these are two very important features which decides the root architecture, and which in turn decide how much plant or the nutrients will be absorbed, because a better root architecture makes the plant sustainable in terms of growth and development, while a poor architecture, it doesn't help the plant, and it is not conducive for the growth and development or to encounter and survive the stressful condition. So this is the project I got through my National Women Bioscience grant. So this was about interaction between glucose and salicylic acid signaling. And in this project, what we could see is that while the glucose could promote the root growth, as it antagonizes, inhibits the root growth, it promotes the stress responses. And finally, we could see at the molecular biology level that happens because glucose and salicylic acid, they antagonistically regulate some of the cell cycle related genes. So these are all cell cycle related genes which are antagonistically regulated by glucose and salicylic acid, and so is the cell cycle or the cell division. So we have proposed a model wherein glucose and salicylic acid, by controlling a common set of genes, they antagonistically regulate each other. Apart from studying the effect of sugars on root growth, we have also looked at the hypothetical growth response, and there are some of the very important parameters which the interaction of the two controls. One of them is the hypothetical length, and another is very beautiful, as you can see in this picture. The hypothetical gravitropism is greatly influenced in the presence of hormones and the glucose. In the presence of resveratrol, which is a plant hormone, the hypothalamus, they lose their gravitropic response, while in the presence of glucose, they just restore it. So that means glucose gives them the ability and tells them that you are a stem, and then you have to move against the gravity. So these are some of the beautiful fundamental features we have been able to explore by using erythropoiesis as a model system. Apart from the growth and developmental responses, we have also figured that the presence of sugars, they can give plants a memory, an ability to remember things just like human beings. So this is a very beautiful experiment where we could show that when the plants are challenged with high temperature or the lethal temperature, then they just simply die, whether they are present in 0% glucose medium or 3% glucose medium. But when these plants, they are given progressive stress, that means first they are challenged with a lower stress or lower amount of stress, and then they are challenged with the lethal stress, then we could find that in presence of glucose, they could survive, they could remember that they were previously challenged with a similar stress, and they have generated certain kind of a memory so that they could better tolerate the lethal stress and they could survive when they were grown in the glucose containing medium. We figured out the mechanism and we could actually show that the presence of, there was some presence of epigenetic marks such as histone trimethylation marks on the histone of some heat stress memory genes, and because of these marks, these heat stress memory genes, they could show a sustained expression even after the stress was withdrawn. So because of the higher expression of these heat stress memory gene at a later time point gives the plant an ability to survive the heat stress better. So apart from this, we were also interested in understanding why there is such a big overlap between the glucose and the hormones in controlling so many growth and development and stress responses. And we carried out several microarray experiment or the whole genome experiments to find out, is there an overlap between the two signal transduction? And we could actually see that there is a huge overlap, almost 62% of the gene in terms of gene expression, they were commonly regulated by the sugars as well as the hormones. We started to characterize a few genes so as to find the nodal molecular factors which lie both beneath the glucose signal transduction as well as the hormone signal transduction. So we picked up one of the genes which was expressed very highly in presence of glucose as well as in presence of auxin, and we started to characterize it because this gene, even though it was expressed protein, its function was not known, and it just contained one domain which was domain of unknown function. So it made us all the more interested in this gene, and later on we found that this gene is very, very similar to some of the ring finger estates containing protein. So we named these genes as FLJ, and to our surprise, almost all the members of this gene family, they could interact with SNRK. I will reiterate here again that SNRK is the protein which gets induced when the plant faces stress and when there is very less energy available to the plant. So we started to look for the phenotype for these mutants, and we found that the mutant phenotypes look as if they are constantly under challenge. This is my wild type plant, these are the mutant plants, and they look as if there is less energy or as if they have more SNRK all the time. So we looked at the SNRK levels, and we actually found not only the level of SNRK was high, but also the activity of SNRK was very high in these mutants. And as I told you, whenever SNRK is more, it blocks the activity of TOR kinase. We could actually see that yes, the activity of TOR kinases was very, very low in these plants. Since these genes, they were also affected by several of the stresses, we looked for the stress response of these mutants, and we found that they were resistant to stress. So we have proposed a model that this gene family member, it acts as it maintains a balance between the activity of SNRK and TOR kinases, and it helps the SNRK and TOR to decide whether the plant has to follow a growth and developmental pathway or it has to go for a stress response pathway. So they are the master regulators in the plant. So basically the conclusion of my work, which I have done till now, is that a role of sugars, a role of glucose and its interaction with plant hormone, it is very, very important for controlling seedling growth and development. And we have also figured out novel signaling regulators of sugar signaling, and which can be modulated and which can be changed in such a way that the stress responses of the plants can be regulated. And these findings can be used from the prospects of biotechnological point of view. So as I was requested that I should let you know about the contribution of Janki Amal Award on my career. So I would say this was the first recognition which was bestowed upon me. And before that, I didn't win any awards, even though I applied for so many of them. So I was first recognized by this award in 2015. And once I got this award, and then there was, I got recognized from various academy. I was recognized by JCPO's fellowship. So I would say that was the starting point that gave me a recognition in the scientific community. So it really helped with my career. It helped me with better networking. As the previous speaker said, the women, they get less opportunity because of their dual responsibility to network, to attend conferences, and to interact with the fellows, with the peers. But once you are recognized, it's very easy to make a mark and to network with other people. And it is very important to say that it gives me financial capability to run the lab smoothly for the last 5 years. And these years had been really, really very tough because of the corona. We didn't have like the grant release in time and all. But because of this grant, I could use this money to support my students who were in the final year of their PhD. So I could give them SRF fellowship so that they can complete their work and they can work in my lab till they get a postdoctoral opportunity. So that way, the grant had been really good for me. With the grant, I could sustain my lab for the past 5 years. And not to mention the personal award money, because we all have ambitions, we all have, we think in terms of like what can we do more for our kids and family. And with the personal award money, of course, we can definitely do a lot. So thank you so much, DVT, for giving me this award. And with this, I would like to acknowledge all my students, all my colleagues who have helped me to reach the place I have been. And I thank NITGR, DVT, and DSD for giving me the grant and the support and the encouragement. Thank you so much.
Thank you, Dr.
Yeah,
Can
yeah.
you stop sharing your screen, please?
That's what I am trying to do. Yeah, yeah.
Thank you.
So I'll be happy to answer any question if there are any.
We don't have any questions right now. Thank you, Dr. Aishwarya. We now
Thank
move to our next speaker, who is Professor Sharmishna Banerjee. She is a professor in the Department of Biochemistry, School of Life Sciences, University of Hyderabad. She teaches immunology and enzymology, and her research focuses molecular pathogenesis and immunology of mycobacteria, HIV, and mycobacteria HIV comorbidities. She is recipient of several national awards such as INSA Young Scientist Award 2005, BioAsia Innovation Award 2008, Innovative Young Biotechnologist Award 2011, and Janki Amal National Women Bioscientist Award 2016. Over to you, Dr. Sharmishna.
Thank you so much for that introduction. So that's okay. That's okay. Absolutely okay. Yeah, that's absolutely okay. Can I share my screen, please?
Yes, I will just make you the host. Yes, please. You should be able to do it now.
Okay. All right.
Dr. Sharmishna, can you also switch on your video, please?
So I did try and it said you cannot. So let me just see. Let me just see.
Please put your slides on display.
Yeah.
Display mode, please. Full screen.
Okay. So is it visible?
Yes, please.
Thank you so much for having me here, and very warm wishes and greetings on the occasion of International Women's Day to all of us here and all over the world possibly. It's a pleasure to be speaking here on this occasion, and I'm also feel very honored to be one of the recipient of Janki Amal National Women Bioscientist Award. So today I'm going to share about what this award recognized in my lab, and it has been a tremendous encouragement to our lab, especially in trying to explore a new area which we had not earlier started, related to mycobacteria HIV co-infection. So my laboratory has been working on two human pathogens, Mycobacterium tuberculosis, the TB causing bacteria, and human immunodeficiency viruses. But this award was a recognition given to us for our work that we initiated on understanding HIV mycobacteria co-infection biology, and also for developing a very simplistic lab model that permitted us to support high throughput investigations to identify new drugs, new targets, especially for very customized for co-infection conditions. So without just trying to give a perspective, tuberculosis and HIV we know is a global health problem, and TB is the most common infection which constitutes 60% of the mortality under the co-infection stage. And the reason possibly is that the very immune cells which are responsible for containment of TB are the ones which are under attack in HIV patients. India has a concern because it has highest number of both the kind of diseases, and very recently has been in a very long list of the top 20 countries which has this looming problem of co-infection, MDR, TB along in both the tuberculosis as well as in HIV co-infected patients. So these co-infections are a concern not only for mycobacterial pathogenic mycobacteria, but during our research as well as others have noted that there are a lot of non-tuberculous mycobacteria and environmental mycobacteria which otherwise are seemingly harmless can also co-infect HIV patients. Concerns are that they remain asymptomatic, they cannot be treated by conventional TB, overburden and cytotoxicity of the drug is a concern, and that actually prompted us to look into that we have to look into the co-infection biology in order to find out that if we have very specific targets for co-infection, especially in the context of mycobacteria in HIV patients. So we started our studies with a field observations spanning around 250 subjects, and for the first time kind of proposed that there is a huge discordance that we generally see in CD4 T cell levels with the viral load, and proposed that non-correlation of CD4 T cell levels and viral loads in the co-infection background possibly should be taken very seriously, because we cannot use these parameters for scoring the disease progression. Not only that, we also kind of this field observation led to certain immune markers or immune pointers that whenever that can give us some understanding about what is the possibility of increased susceptibility to hyperinflammation when there is a co-infection case, or can be a possibility of relapse associated with the HIV cases. Also, we have noticed that there is a huge prevalence of atypical mycobacteria, environmental mycobacteria. All this prompted us that if we require to do a very high-end cell, understand the cell biology, molecular biology event, we require to have a more simplistic model, and we tried to take all these observations from the field to generate a field compliant cell culture based model which will mimic the readouts from the field. So this model was based on the cell culture of macrophages, and we made a model where HIV background gets a secondary infection of a mycobacteria. The model was validated the model can be used not only for pathogenic mycobacteria, but it has been validated for clinically relevant opportunistic mycobacteria that exhibited increased persistence during co-infection together with increasing in HIV titers, and has been glad to recognize that it has been used in certain modified forms or directly by several labs across the world. With the hypothesis that the cellular environment, which actually is the host environment, is nothing but an expression of, can be an expression of differential mycobacterial proteome that is expressed during the co-infection stage, which possibly leads to a very conducive environment for HIV, because of which HIV may be having a better propagation environment. We did a very thorough, so we did this, we used this particular model, isolated and tried to understand the mycobacterial proteome or the secreted proteins either directly into the cells or within the microenvironment, where we also noticed that there can be a cell-to-cell communication through exosomes, cytokines and many other factors, and try to understand that what are the changes that takes place in the host proteome vis-a-vis the intercellular mycobacterial proteome. We did a very intense, dissections of the various pathways, and try to understand different kind of critical host parameters that are altered during the co-infection, to get a background that what is the environment that HIV prefers to live in, and gets a boost in propagation. Just to kind of give a glimpse that we found the many of the usual players which actually reinstated and strengthened our confidence in our model, where we found that the host environment is compromised for endosomal recycling, but more important, even the cargo degradation. Some unusual player, of course, we saw that there are metabolism which is altered, but there are very unusual players like carnitine O-palmitoyl transferases, levels of carnitine which had already been reported to be conducive for HIV, metabolic rewiring in mitochondria, dysfunction of mitochondria, high energy state, yet there are certain kind of receptors that were upregulated during co-infection, which can be used by HIV to have an entry other than the CXCR4 and CCR5, which are commonly known, not to list the various kind of transcription factors and regulators which are possibly pro-HIV activities that were upregulated during mycobacterial co-infection. The question next definitely was, what is happening to the mycobacterial proteome? And we did see that mycobacteria also, and in a single infection versus the co-infection, have a very different set of proteins to express, and also our interest was also in several of the secreted proteins. So we did an analysis on that, and also tried to validate those mycobacterial proteins, the impact of those mycobacterial proteins on the persistence and on HIV titers, using certain mutant bacteria, and try to see that whether such kind of factors can have a say in persistence of mycobacteria itself, even during mono-infection, and what happens to the viral titers when co-infection occurs. Just to give an example, to give a perspective, we do see that one of the factors like IDR, which is the global RN-dependent transcription regulator, it not only is a very important regulatory factor, but it has a huge network of proteins that it can regulate, which shows that had an impact on viral titers. So by doing that, we have listed this particular study has given us a platform, a baseline information, and a lot of mycobacterial factors and the pathways associated with which are the possible or potential regulators, which if we can control, if we can understand, if we can target, possibly can control both the bacterial titers as well as the viral titers during co-infection. To take you, just to give an example that how are we taking this study further is, right now we are working with, after getting this enormous baseline information, we are working with the hypothesis that mycobacterial proteins that are released either within a cell if they are co-infected, or within the vicinity of the cells which are maybe independently infected but are influenced by each other due to released or secretory factors, can happen in two ways. Either the mycobacterial proteins can go and directly interact with the HIV-LTR, thereby promoting viral transcription, or they can also influence the host and make the host very conducive for viral propagation. It can induce certain factors in the host which actually are the positive regulator of HIV propagation. One such example is our earlier observation on co-infection, where we had shown that one of the host factors, which is a zinc finger protein, which we established as a positive regulator of HIV-1 LTR. This host factor goes and binds into HIV-LTR. We have characterized it intensely, how the binding takes place, and if you see in the HIV co-infection background, there's a tremendous expression of such kind of host factor, which the virus can then recruit and propagate. So we found now with this study that there is a mycobacterial factor which is responsible for this, and this mycobacterial factor can drive the expression of this protein ZNF134, which kind of tells us that while we were observing earlier a tremendous increase in this ZNF134 factor in TB patients. So our hypothesis for many such factors that we have listed now is that mycobacterial infection, whenever happens alongside HIV, results in upregulation of such kind of positive factors like the ZNF134, which actually is induced by or played by mycobacterial factors like MTRA, and that results in increased LTR activity and hence the formation of HIV, and therefore we have high viral load in HIV patients with TB infection. So this is kind of not only maybe true, maybe indirectly or directly there are mycobacterial factors that are responsible for it. So towards this, we are trying to, validate a number of factors, and for all of them, so yet another factor is ESPR, again an MTB virulence regulatory factor, which possibly does it through indirectly by regulating the IL-4. We try to show them that they do migrate to the nucleus to have this effect. They bind to the promoter region, and several kinds of these validations in place. So I'll be summarizing the hypothesis that we are working on with the help of, in support of this particular program, and there are a number of such factors which we feel that can be potentially regulated. We have to make us understand the co-infection biology, where we feel that there are several factors that we have identified that are secreted directly into the cell or are in the vicinity, and these factors, they find their way into the nucleus, and we see them in the nucleus, and when these factors, they bind to the LTR of HIV, or maybe indirectly, it tends to increase the viral transcription, and that results into increased viral titers. So our right now aim is to inhibit all these stages by which we feel that these mycobacterial factors are helping, and if we can manage to inhibit these interactions, these vibrations, or the secretion process, maybe we will be able to restrict the viral RNA transcription and resulting in controlled viral regulation even in the co-infection background. That may also helps us in designing drug molecules which can be used in co-infected conditions, and not controlling the disease progression, which otherwise would take place if it is not controlled during co-infection. Yet another approach is that we have seen that there is a thorough abuse on mitochondria during the co-infection, and we are in the process of screening and actually have come across some of the small molecules that improves the mitochondrial health, and we are in the process of trying to see that if such kind of molecule can be used alongside the therapy that is used for co-infection, whether that will help us in further increase mycobacterial clearance or decrease in the viral titers. So with this, I acknowledge the patients, the clinicians and medical staff, all our collaborators. A number of people helped me in arriving at where we are, but the recognition through Janaki Anand National Women Bioscientist Award has been a tremendous boost in the lab, because it has, we have now a set of, I will call them budding research scholars who are so interested in understanding the co-infection biology that we believe that at some point of time we would be able to not only understand the co-infection biology, the molecular and cellular event, but take it to a translational level. Thank you so much for giving me this opportunity, and thank you once again, CPT, for recognizing my work through this award. Thank you so much.
Thank you, Dr. Sharmishan. We now move to our next speaker, Dr. Lakshmi Prasanna. She is a biocare awardee, and her project contributed to development of a novel chitinase enzyme-based technology and consolidates the industrialization of high-end biopesticides and biocontrol products, which should provide technical support to biopesticide industry. The technology pertains to a recombinant chitinase enzyme and is protected by granted patents in US and India. She is now exploring opportunities in industry for tech transfer. Over to you, Dr. Prasanna.
Good afternoon, all. At the outset, I would like to thank Dr. Suraksha Diwan and DVT for the invited talk. My biocare project and its research outcome, which led to an enzyme innovation, is focused on the development of 1 minute. I'll just. Yeah. Yeah, can you please help me with the share screen? I'm not able to share the screen.
Dr. Lakshmi, I've made you the co-host. It should allow you to share the screen. There is a green
Yes,
button.
ma'am.
Yeah. Okay.
Yeah.
Yes.
Is it visible now?
Yes, please. Yes, you can start.
Yes.
Yes, thank you.
My Vigate project and its research outcome, which led to an enzyme innovation, is focused on the development of a novel chitinase-based enzyme technology aimed at a breakthrough in integrated pest management for a safe and effective sustainable agricultural practice and other possible industrial applications. The motivation for the Vigate project proposal is my tryst with a new chitinase from a novel strain of Brevibacillus electrophoresis based on the preliminary research carried out at Norwegian University of Life Sciences in collaboration with Professor Vincent, a pioneer and world expert in chitinase research. Bicare funding gave me a powerful opportunity to develop a new proprietary technology and an enzyme innovation in biopesticide and chitinase research driven by UN Sustainable Development Goals with an objective to increase the competitiveness of biotech industry in India and overseas. The primary goal is to improve the sustainability of Indian agriculture industry and contribute to the forestry area. The problem is most of the currently available biopesticides have been limited to Bacillus thuringiensis, and majority of the pests have developed resistance to Bt-based biopesticides. So the aim of my research project is to discover and develop new biopesticides with improved stability and efficacy, which will have a reasonable opportunity for being commercially successful with unique benefits that current products, in particular Bt products, lack. So then the solution that the Biocare project aimed at was at establishing a chitinase-based technology transfer platform with the ability of continuous innovation for a new generation biopesticides and biocontrol products and as a possible alternative to Bt. So why did I choose Brevibacillus electrophoresis? So a biological cousin of Bt. So the Brevibacillus electrophoresis is a rarely distributed emerging entomopathogenic bacterium. So as I mentioned, it's a biological cousin of Bt and Bacillus sphaericus. So the isolated strain is a hyperchitolytic one and is a rare find. So novel chitinase is a robust and high performance enzyme with significant traits like marked thermostability and alkaline tolerance. And the most important and promising research finding is that the chitinase can provide a solution to chitin waste management by effective industrial scale utilization of environmental chitin like waste, marine crustacean shells. Yeah. So the research premise is the first and foremost goal of the research premise is development of an integrated research platform for a novel proprietary technology, mainly for biological control. But when I was performing my experiments, I could just find out that it has many potential industrial applications in the areas of bioremediation, biofuels, etc. And so the chitin waste management can lead to the enzymatic production of value-added products. So like N-acetyl glucosamine and D-acetyl glucosamine, which have highly medicinal value. And so I have just listed out like a few unique characteristics of this chitinase. So like as I mentioned, it's like alkaline tolerant and which is active over a broad pH range 3 to 11, and the optimum pH is 9, and it is thermoactive. So again, it has a dual activity, and the exochitinase acts at 55 degrees centigrade, and the endochitinase activity was exhibited at 65. So and it's highly thermostable with a range of like 70 degrees centigrade, and it's an extracellular chitinase which has high affinity to natural substrates like shrimp shells and crab shells. So a recombinant chitinase is secreted into the extracellular medium, making it suitable for large-scale production required for all the industrial applications. So I could devise a novel purification protocol using powdered crustacean cells, making the large-scale purification strategy very simple and effective. And then lastly, the dual activity of the chitinase makes it very efficacious, and it has a lot of benefits over BT pesticides, because it can be used as a contact and ingestion biopesticide as well. And just to share this, Novozymes is a global leader in enzyme research, and they have announced in early 2021 the collaboration with FMC, which is a US-based agricultural sciences company, to work on enzyme-based biocontrol technology. So I would like to just mention that I have filed my patents in 17, Indian patent in 2017, and US patent in 2018. So much before Novozymes ventured into this biocontrol technology. So, I mean, this needs to be acknowledged by the scientific fraternity. I've got my granted patents just 2-3 months ago. Indian patent was granted in December, and US patent in November. And so I would like to conclude my talk with just mentioning a few points. So CCMB hosted my research work for 4 years. I joined CCMB as a research consultant to support Indo-Australian project on lipases. And then I continued with my biocare project. So I'm an army wife who had career breaks because I was into work-life balance. So since I'm very passionate about contributing to research, so whenever I got an opportunity, so I could work and contribute and try to be productive. So now I'm just trying to seek technology mentorship and then seeking grant funding also. And I'm trying to build partnerships with the help of BIRAC, DBT, and BCIL for scaling and refining the technology, and for the technology transfer to different companies. So I could list and I could identify and list the companies and share it with BCL and BIRAC. So I will take it forward from there. So I need to understand the landscape to foster the innovation, target market, and customer segment. So, and I also need to collaborate with technical expertise to continue innovating and finding new applications. So probably I would come back to DBT for some kind of support, either this grant or because I don't have a permanent position, so I need to associate with an academic institute, an academic lab, or an industry, and then would like to develop the product. And I deeply acknowledge the support extended to me by Dr. Suraksha Diwan, Dr. Renu Swarup, Dr. Nitin Jain, Dr. S.R. Rao from DBT, and the present and past directors of CCMB, and Dr. Archana from IP Group, and my group leader in particular, Dr. Madhusudan Rao, and all my lab mates for all the learning and fulfilling experience to make this work. Thank you all.
Thank you, Dr. Prasanna.
Any questions?
We don't have any questions right now. Thank you for your talk, Dr. Prasanna. We now move to the next speaker, who is Dr. Anandi. She is a Biocare awardee, and her funded research was on quantitative tissue proteomics of oral tongue squamous cell carcinoma for novel biomarker discovery. She was a faculty at RDR Cancer Institute, Chennai till very recently, and has founded a company, Clin Biocare Technology, where she is also the CEO. She is also director of Saral Biosciences Research Institute. She is passionate about her new endeavor, and she has been working with medical students, undergraduates,
for the kind introduction, but I couldn't share my screen.
Oh, okay, give me 1 second. He's trying to.
Yeah. Is it visible now?
Can you put it in the slideshow mode, please? Yes, this is fine.
Okay. Thank you. Thank you for giving me this opportunity to present about myself and my company, my Biocare grant and the contributions. So myself, after my master's in Madi, I've been a hospital scholar in Madurai Kamaraj University, where I pursued my doctoral studies with proteomic technology. And further with that, I was trying to explore more, so I want to move on with different field of expertise. So I moved with the IIT Madras, Indian Institute of Technology Madras, for my PhD, where in that period only I got this Biocare award for and with that grant I was able to move to Adyar Cancer Institute as I was in need of more number of cancer samples. So my career started as a microbiologist and landed as a faculty in Adyar Cancer Institute. For this transition, DBT Biocare award was really a great milestone. With that thing, I want to do more, so I just quit my faculty job and I landed in creating my own company. So I'm leading now 2 different biotechnology research companies. If you words about the grant what I obtained from the especially tongue cancer. The need for the study was when we look into the literatures, you're not having much reports with Indian populations, and there was always an devastating about this oral cancer as it has been diagnosed in a very late stage, and we need emerging markers which can tell us the condition of the patient in very early stage. That is the only option where we can save lives. So with that information, I just started by having the approaches using 2D dyed electrophoresis coupled with mass spectrometry to identify the markers. Not only identifying, I want to characterize and I want to validate them with more number of samples. And ultimate aim of this project is to create a very easily available assays using salivary samples where we can diagnose the cancer patients in the early stage rather picking them in a very late stage. This is just an information what I tried with the samples, and this is an image describing the differential gel electrophoresis using one gel for comparing both normal tissues as well as the tumor tissues. And with this, we are able to identify about 122 differentially regulated spots, and the marked region shows you the clear difference. It's from the same patient. Normal tissue was the adjacent normal and the tumor tissues, and this technology was able to pick up most number of differential regulated protein spots. With that, we did multiple gene ontology analysis and multiple gene ontology assays to find the novel markers which are very specific to our Indian population setup. And we derived with about 24 candidate markers which were having a significant differential regulation. And apart from identification of discovery phase, we went for validation phase using qRT-PCR, and we found a similar trend happening in RT-PCR like expression level, also in mRNA level. So we carry forward with this 24 markers for further analysis of our work. As I was mentioning earlier itself, we need markers specific to Indian population setup because it is not the same. So we can't use the markers which has been already available with the Western population. To confirm this aspect of concept, we did a bioexpress analysis where we took the data which is already available with Western populations and compared with the data of our Indian population setup, what I recently did, and we found 6 genes to be having strong negative correlations, which means these markers are totally different and unique to our own Indian setup. So we need Indian biomarker-based studies to have a better treatment option with respect to cancer discoveries and cancer drug discovery aspects. So apart from qRT-PCR validation, I went for Isc analysis as well. In this, we have done about more than 10 markers with different stages of infection, different stages of cancer starting from 0 to 4th stage. And we found all these markers have a unique character which been having a highly upregulation during cancer progression. And apart from this, we took 4 markers based on the proteomic study, RT-PCR, IHC validation, and we went for salivary ELISA. So we took the salivary samples from normal patient, like normal healthy volunteers, and we took from an IP going for a cancer stage, and we took for cancer samples as such. So we want to evaluate all these markers pattern, how it being picked up in a very early stage, and we found that certain markers like vimentin and other proteins have a capability to be picked up in a very advanced, like a very early stage than going on advanced cancer progression. So with that, to summarize about the work, what I did with particularly DBT Biocard support is this is the first study to use this 2D DICE technology using an Indian patient samples, especially tongue cancer, because we have more reports with other cancers apart from tongue cancer. So we were able to identify novel markers which is specific to Indian population, and not only identification, we went for a validation. As stopping with that, we aim for an assay development with especially salivary samples, and we have got identified all these 4 candidates like vimentin, S100, A11, cystatin B and galactine-7 are being highly promising, which can be used further as a marker to detect this cancer in a very early stage. And I've been in process of continuing this study in with other collaborations as well, though my IPR funding is complete. I have taken these basic information. I'm trying to do a technology transfer with other companies where we do the strip development. The specific points I want to mention about DBT in my career, it helped me to develop multiple networking, and it gave the first scientific recognition as an independent researcher. And on the whole, it influenced me a lot. So I had my company named Clin Biocare because of this biocare thing. I was being recognized in multiple levels. So on the whole, it gave confidence to start my own company, and APJ sir is always inspiration from during our having the discussions, and he is the one made me sit back in India and think about Indian problems and Indian aspects. So all these things made me start my own company. And my own company in my own native, so because I am from very much down knowledge in other things, so I have started this company to help my own native people who are studying here, especially girls, and I've been having my own students here being employed, and we have been taking them in multiple level of PhD works as well. So with this, few words about the company, what I've been working with. So we work with cancer biology, microbiology, marine biotechnology, human diseases, plant biotech, and reproductive biology. And especially we focus with the more student aspects rather with more of this other projects in them, and to connect the gap between merging with the academical things so that it can be translationally converted to patients who are in need of this biotechnology requirements. So these are few of our recent activities which I was doing with multiple people, and these are our few clients already being in touch with CBT. And thank you for giving this wonderful opportunity. I welcome you all to such company. It's about 3,000 square feet company where you are getting employment and you are being trained and you are giving multiple research aspect projects. Thank you very much for this opportunity.
Thank you, Dr. Anandi. We now move to the last speaker in the series, but by no means the least. She is Dr. Bushra Alvi. She is an associate professor at IIT Kanpur, and her research interest involves exploration of genetic and epigenetic changes that initiate cancer and its progression. Apart from many other awards, she holds the DBT Wellcome Trust India Alliance Senior Fellowship 2020. She was also awarded Ramachandran National Bioscience Award for Career Development from DBT 2020 and Shanti Swarup Bhatnagar Prize 2020. She has been recently named among the 75 under 50 scientists shaping today's India in a compendium issued by the ST Government of India. She'll be taking us through her scientific journey today. Dr. Bushra.
Yeah. thanks, Surya. Like, can you please make me host?
Yes, I'm just doing that.
Yeah. So, thanks to DBT and ICGB. Yeah. Can you see my slides?
Yes.
Yeah, thank you. Thanks, Surya. So yeah, I'm really indebted to DBT and ICGB for giving me this opportunity to talk on this event, and especially to Dr. Diwan and Dr. Dhruv. So I was told to talk about my journey, and my philosophy about journey is if opportunity does not knock, build your own door, in brief. So I belong to a small town, Bareilly, and I did my high school from there. And after that, I was admitted to Aligarh Muslim University for my senior secondary school, and I pretty much finished my entire education there. So I did my SSC and BSc, MSc, and PhD in genetic toxicology from the same institution. Since my PhD work was more focused on genetic toxicology, it's more about studying the effect of herbicides and weedicides on fish or aquatic animals. So I used to read a lot IRC monographs, and that incited interest to do cancer biology, because most of these chemicals, they have some oncogenic effect. They are mutagens, some are potential to be a carcinogens. So with that dream, I moved to New Delhi and joined Dr. Neeta Singh's lab at AIIMS in Department of Biochemistry, and there I started working on a DBT-funded project that was on identifying some oncogenes in lung adenocarcinoma. And subsequently, I also received CSIR Research Associate Fellowship, and I moved to NII. And with that brief stint, I received another fellowship that was like a training research grant from Canadian Health Institute, and that exposed me to more of a cancer biology in prostate cancer and breast cancer, and I studied their epigenetic alteration in these cancer types. At that time, that was a time around 2000 and 2007 and 2008. That was a time more and more people are moving towards from microarray to next generation sequencing. So with that hope that I want to learn more about NGS or cancer genomics, I moved to University of Michigan, and there I was I had a couple of fellowships, including Genentech postdoctoral fellowship. So that stay was a little bit longer, and at the same place I also served as a research investigator for almost 1.5 years. And around 2012, I decided to move back to India because I thought I had enough of training as a postdoc, and I was fortunate to get the fellowship, intermediate fellowship from DBT India Alliance. That time it used to be DBT Wellcome Trust India Alliance. So so I got fellowship first, and then I got the job at IIT Kanpur, and I established my own laboratory in 2013. And currently I'm a senior fellow there. So my group focuses mainly on cancer biology and other aspects of cancer, including cancer genomics. So again, why I'm in this field? One of the reasons is it gives me freedom to do, to do the work I want to do or choose a problem of my choice. Besides that, it creates a lifelong impact on the people who work with me, especially my students, my mentee. So as a child, I was a curious child, and most of the time, I used to catch butterflies and watch fireflies, catch them, put in a bottle, do all sort of weird stuff. So always the thing is why it's happening. The question is to ask okay, why they glow at night. So with that, there was a scientific acumen in the background, and with this, I developed interest in biology and other sciences. And after doing my 12th, SSC, I decided to go for as usual for a PCT student. The dream is to become a doctor, so I decided to become a physician. And some of the traits that really impressed me was how empathetic and how confident they are, they're going to cure the diseases. But unfortunately, I could not get through that. And I don't want to be a person who will work on a BDS or more of a dentist. So I decided to move on with my higher education. After BSc, again I started to think what I want to do. Then I thought okay, with all the patriotism, I decided to become an Air Force officer. And that time I went to this place Varanasi. They have short service board, it's a small Air Force base. So I qualified written exam there, but again I failed in their physical test, that is like monkey crawling and the rope climbing. I failed. So again, because science was my passion, I moved on. I said, okay, I'll do my higher studies. I went to do on PhD. But if you look, most of the trades in these things, whether it's a physician or an Air Force officer, is there. So I decided to become a scientist when I started my PhD. So currently my lab focuses on, my group focuses on understanding what initiates cancer and how it progresses, how it moves to other parts of the body, what we can do to control it. So in a nutshell, that's what we do. So pretty much other important aspect of being in this position is still I retain my this curious childlike attitude. So again, I'm going to ask these questions, but in a different way, in a more mature way. So again, so when I decided to join IIT, I was having few other offers as well, but then obviously you can think about, I want to do biomedical research, I want to work on cancer, and joining IITs means hey, IITs are known to develop technologies, we recently even developed this ventilator and even oxygen concentrator and robotics. And the big challenge was okay, from where I'm going to get clinical samples, because for cancer research I have to do if you really want to make an impact and take your research to the patients you want to work with clinical samples. So in one of the meetings, I asked one of my friends who is working in one of the cancer hospitals to help me get the clinical samples. So his answer was okay, you move to IIT Kanpur, it's very difficult to get clinical samples. You better work on cancer cell lines. So to all my colleagues or women or other students or whoever is sitting there or listening to this, don't take these kind of suggestions, don't take no as an answer. So I started exploring this local area and established a good niche of collaborators within Lucknow, Delhi and Kanpur. Now we also have collaborators in Tata, Mumbai. So that was the biggest challenge. And then obviously in India, the follow-up is not that good. So usually you get the patient sample, but then what will happen with that patient after 5 years, after 10 years, we don't know. So that is very important. And then how to team up with the pathologist to get the clinical details. Then other the roadblock was how to get samples in a good condition. So we have to create pretty much our own cold chain, get the portable small liquid nitrogen cylinders, carriers, and get the samples in a good condition. Obviously, when you want to do cancer biology, immunodeficient mice facility is must. And at that time at IIT Kanpur, there was a mice facility, a temporary, but not the immunodeficient mice facility. So I started from the scratch, and it was in the middle of nowhere, and in a small room I started. And at several occasions, the mice were attacked by a meerkat or a rat, a wild rat. Rats will injure and they eat up the mice food. So now we have a very nice, sophisticated 5-story building there in the campus, and this problem is resolved. Not only that, the IIT Kanpur, they decided to start the School of Medical Research and Technology, SMRT, which is designed by the Horseback. These are the leaders in setting up the hospitals. So eventually, things will pan out once you have a, will, how to, if you have perseverance, you know how to chase it and achieve it. So my my group focuses on these cancers. We primarily work on prostate cancer, colorectal cancer, breast cancer and glioblastoma, and we use array of technology like cancer genomics, fluorescent in situ hybridization, and other functional genomics aspect of the cancer biology. Try to understand the mechanism behind the oncogenes and how they evolve, and also we try to look for some of the drugs which may be which could be used for targeting those pathways. So in pretty much we ask what is the molecular mechanism, what is the basis of drug resistance in many cancer, because many cancer usually they respond to cancer drug or chemotherapeutics which have been used, and then over a period of time they develop resistance. So what is the mechanism of resistance, how we could overcome. We also use cancer genomics to explore new cancer biomarker and genetic aberration in Indian cancer code, especially in prostate cancer, and finally translate all these findings into precision medicine to help patients. So this was the first study, like, so I joined in 2013 and I tried to, like, collaborate with several hospitals around Kanpur. So within Kanpur, we have GSVM hospital, we had collaborators from there, AIMS New Delhi, as well as KGMU, King John Medical University, Lucknow, and even a private pathology lab in Gomti Nagar. So Dr. Vinny Tandon, who is on this paper, also like collaborated in getting all the sample. And this was the first characterization of the prostate cancer patient from India, and what we found that pretty much these statistics were a little bit different. TM+2R, which is a highly recurrent genetic alteration in Caucasian population, was also similar in Indian population. Beside that, RAF kinase alteration was more. So in Caucasian is only 2%, whereas in Indian it was found to be 6%. And so this was the first study to show these are the some of the established alteration prevalent in Indian prostate cancer patients. So currently what we are doing, we're exploring the mutational landscape of Indian prostate cancer patient, and we have several collaborators, clinical collaborators, as I mentioned, including Tata at Mumbai. And what we from this pilot study, what we did, we sequenced few patients, and what after analyzing the data, the whole exome sequencing data, we found more missense mutations, and the average mutation rate was around 2.37. So usually, for advanced stage patient, anything more than 1 is supposed to be the patient is having much more advanced stage disease, and that is one of the reasons why we got a little bit higher mutation rate, because most of these patients are diagnosed at late stage. So early detection is very important, and this is one very interesting lead that we got from this patient. We found that one patient he had this CDK12 biallelic inactivation. Biallelic inactivation of the gene, and because of this inactivation, these mutations are in kinase domain, and because of this, you see there is tandem duplication in the entire genome. And because of this tandem duplication, we see a lot of new antigens. So it's known as a new antigenic burden in this patient, and this makes these patient much more susceptible to immunotherapy. So our lab is our group is also working in understanding the pathobiology of CDK12, biallelic inactivated mutation positive patients, and what therapies could be used to benefit these patients. Now, because today is a very special day, so I thought we should put this slide, and I'm really indebted to DST, especially this Vigyan Jyoti scheme, and it's a wonderful scheme, and I'm sure DBT will also come up with similar strategies. So they provide 3 weeks residential camp for the brightest class 11 girls, and we had them, I think it was in 2018, and it's a residential camp, and they go to different labs, they do hands-on experiments, interact with the faculty and the other students. So this was a batch that I mentored at that time. So you can see how bright and beautiful these girls are. They are full of questions and full of enthusiasm, and if given right opportunity, I'm sure all these girls will eventually become leaders in their field. Not only the women who are there in this picture down here in this Women in Science and Engineering conference, but they have potential to even become Elizabeth Blackburn, these Nobel laureates, or Barbara McClintock or Marie Curie. So I think we have to give them opportunities and encourage them and give them wings to fly. So now I'll end, this is my group, little bit old picture, but I'm blessed to have such a motivated and dedicated students here. And not only that, last but not the least, the funding agency, India Airlines, and DBT, as SERV, and especially recently I got this SERV Power grant. And I'll end my talk with this quote, which I like a lot. So our deepest fear is that we are powerful beyond measure. It is our light, not our darkness, that most threatens us. We ask ourselves, who am I to be brilliant, gorgeous, talented, and fabulous? Actually, who are you not to be? You are a child of God. Your playing small does not serve the world. So thank you very much for giving me this opportunity.
Thank you, Dr. Bushra. That was a very touching end remarks. Now I invite Dr. Isc Analysis, group leader and coordinator of Biotech Review, ICGP, to give his closing remarks.
I think just before we go for the vote of thanks, I think if audience has any questions, because in between we were not asking because of the time and other factors. So I wonder if others have any questions, then they can come forward before. And I have just one question to start with Dr. Anandi Sivaganam. Is Dr. here?
Yes, sir.
Okay, so just one quick question, because before the floor is open to that. You started your career with a BioCare project in the R&D, and I can see that later on you moved into a startup and you started your own company. So can you just tell us
Yes,
a little bit more, not about the science but about your own mental process, your thought process, that suddenly you change the course of action of your career from a researcher to an entrepreneur, because what has happened, what was thoughts which were coming into your mind, that is one. And then, I can imagine being a startup, and that too a women startup, then will have a lot more challenges. So can you just give a little bit, there's one challenge, and how did you overcome that?
Yes, sir. Thank you for asking the question, sir. I have faced a lot of challenges in executing this startup and things, but the thought process came because of this DBT Biocare. I have been noted by many people as a bioproteomic expert. So people started asking me about second opinions on proteomic technologies and proteomic aspects, and we are not having much people working with this gel-based approaches with mass spectrometry and other things. So when I was helping others, and many people asked me, why don't you start this as a separate service like division, where many will be getting the help from you, and it can be reaching more people. So that is the first point. With the influence of DBT Biocare, I was able to know the lacking area where we can pitch in, and I can use my expertise of this 18 years, where I adapted this technology from my PhD days and use in my postdoctoral studies as well as in my faculty job. So that is the area I started. And while establishing this startup thing and major investments and other things, it was really a major challenge. And exactly when I started this thing, we got Corona as well. So we were having a lot of disappointments and a lot of things, but we were fortunate to sustain the situation, and we were getting people support, and we are getting more students coming in from the colleges, as they don't have facilities in their own college, and they come up and they were expecting to have hands-on training, where I could help them with my small setup when I started. Even I remember when I was starting, I was having only 2 tables, workbench tables, with one small instrument setup for my proteomic technology. And from there, now I have built up about this 3,000 square feet of complete setup with accommodations only for girls. So I would like to help more girl students in reaching these, especially in science wing, because I have faced a lot of politics and a lot of downs in my career when I started. So that is the main thing, and main inspiration is to help people with my knowledge, how much I can reach. For that thing, I feel startup is the only thing where you don't have any other domination, and where you can work for your dreams and you can run for your dreams and help people in more better way. Thank you, sir.
Okay, thank you. I think anybody else has any question to any of the speakers? Because so to any one of them. And very quick, before anything comes, just to reassure Dr. Bushra Atik, because I'm very happy to see the slide about the Vigyan Jyoti program which you were showing, madam. And the reason is, like, I'm unable to stop myself, because I joined DBT only 6 months back, and before that I was head of the career program and the Women Inside program in DST. Yes. And during that time, this particular Vigyan Jyoti program was conceptualized. Of course, there was some seeds were there, but actual design, the actions, the planning, and that was done. I was very much instrumental in that. So Vigyan Jyoti is one which I am very happy to see that has come to your slide. So if you are happy, and I was watching the nearby this Navodaya Vidyalaya school, Kanpur Dehat, I was in touch. And with their 2 visits also, and I'm aware, so I'm very happy for that part. But in addition to that, I would like to reassure that, not because I have moved, but already DBT has been doing, but in the past I have been instrumental in starting this Vigyan Jyoti. So the program called Gati, G-A-T-I, which is again started in coin in my time. And third, another scheme called Mobility Scheme for the Women, which was again lying for under consideration from last, I think, 7 years, 6 years, which allows the permanently employed women scientists who had a regular job in IITs, in universities, to have mobility because of family or personal reason from 3 to 5 years. So we started that program last year only with 2 women's IDs. So idea was to have proof of concept that it runs. Now from 2 can become 200 and 2000 later on. So I'm happy to reassure
Thank you so much.
because you mentioned about DBTs, so that yes, DBT and
DBT is doing a lot, but little bit more at the grassroots level is also important. DBT is also putting like lot of efforts, like biocare and other like such programs, like even the Janki Award is.
Yes.
But at the grassroots level also is very important to encourage like these college girls.
That is excellent.
I am like the nodal person for IT campus, so thank you for that also.
I know. And in fact, if you ask me, I've got some of the pictures taken from these Vigyan Jyoti girls. I remember from Haridwar area, and they went to the AIDS, and I shared that photograph of the girls sitting with a cardiologist and professor of cardiology, which was in my, because I was so moved to see that picture, how glad the girls were there. I put it in my WhatsApp as my DP for because all the girls were so happy to meet a cardiologist in surgery, and they were class 9, 10, 11, 12, I think, school going girls. Right. And, so I really I really take your point, and Suraksha is also listening here. Thank you. Thank you very much. We can move ahead.
Thank you, Dr. Mishra. Indeed, these are very important programs from DST and as well as several of these programs from DBT. So I hope all of you liked and appreciated this event organized today on Women's Day jointly by DBT and ICGB New Delhi. To conclude the event, first of all, I would like to thank Dr. Rajesh Lokle, Secretary DBT, and Dr. Dinakar Salunke, Director ICGB, for gracing the occasion and sharing their valuable thoughts. I would like to thank Dr. Sanjay Mishra and Dr. Suraksha Diwan for supporting women-centric schemes including biocare, as well as supporting the program management unit at ICGB PMB. Thanks to DBT and offices of Dr. Mishra and Dr. Diwan for their support for organizing today's event. And I thank all the speakers for participating in the event, their presentation. It was indeed very nice to know their success stories. We also thank Dr. Suvira and her team to organize today's event, as well as ICGB's administrative and technical staff for their support. Finally, I thank all the audience here who joined us today. We intend to organize a full-day conference next year and hope to see you next year in person. Thank you all. Thank you very much. We conclude the event for today.
Thank you everybody. That's a wrap from ICGB now. Have a good evening. Bye.
Thank you very much to everyone, including ICGB host and all the audience on behalf of Department of Biotechnology. Thank you very much. And please send your suggestions to Suraksha or myself if you have anything about women-centric programs which we can fix.
Thank you so much.
Bye-bye.
I think I'll get back. Thank you so much for having me here. Sharmishta here.
Thank you everyone. Thank you, Dr. Mishra. Thank you.